In the spring of 2005, a team of paleontologists in Montana was doing something that happens hundreds of times a year at dig sites around the world. They were breaking apart a Tyrannosaurus rex femur. The bone was too large to transport whole, so they cut it. What came out of the cut surprised everyone in the field that day and divided the scientific community for the better part of a decade afterward. Inside the bone, in cavities that had been sealed since the Cretaceous, there were structures that looked like blood vessels. Flexible, translucent, with what appeared to be cell-like forms inside them. The paleontologist leading the extraction, Mary Schweitzer of North Carolina State University, had seen something like this before in other specimens, and had spent years trying to understand what it was. Now she had a fresh sample, and she had a laboratory waiting. What Schweitzer's team published that year in Science was one of the most contested papers in the history of paleontology. Not because the data was wrong, because what it implied was almost impossible to accept. The conventional understanding of fossil preservation holds that organic material does not survive geological time. Bones fossilize because minerals gradually replace the original biological tissue. The organic components, proteins, lipids, DNA, break down through hydrolysis and oxidation, and within a few million years at most, nothing biological remains. A 66-million-year-old bone should contain no soft tissue, no proteins, nothing that could be analyzed as a biological molecule. The Schweitzer paper reported that it did. The immediate response from much of the field was contamination. Modern bacteria, modern proteins introduced during excavation or lab handling. One prominent geochemist published a detailed challenge, arguing that the structures Schweitzer had found were biofilm, bacterial colonies that had colonized the empty spaces inside the fossil long after the original tissue had gone. It was a serious objection, seriously made, and Schweitzer's team took it seriously. They spent the next several years ruling out each alternative explanation one by one. The structure survived chemical treatments that would destroy bacterial contamination. The carbon isotope ratios were inconsistent with a modern biological source. The antibody tests that Schweitzer's team used to probe the chemistry of the material produced responses specific to vertebrate collagen, not to anything bacterial or environmental. By 2007, a second paper in Science reported that her team, working with researchers at Harvard, had used mass spectrometry to extract and sequence actual protein fragments from the T-Rex bone. The proteins were collagen, and the collagen sequences matched something specific. Collagen is the most abundant structural protein in vertebrate bodies. It forms the scaffolding of bone, tendon, and connective tissue, and it is one of the more durable biological molecules known, which is almost certainly why fragments survived inside mineralized bone long after everything else had degraded. The sequences Schweitzer's team recovered were not DNA. They would never be DNA. At 66 million years, every strand of genetic material that ever existed in every T-Rex that ever lived has broken down completely. The theoretical maximum survival time for DNA under even the most favorable preservation conditions is somewhere around 1 million years. Some estimates put it lower. T-Rex is 66 times older than the upper limit. The genome of Tyrannosaurus rex is gone, and it is gone permanently. What the collagen sequences could do was something different. Proteins are, in a sense, an indirect record of the genome that produced them. The amino acid sequence of a collagen protein is determined by the DNA that coded for it. By reading the sequence of the protein, researchers can make inferences about the sequence of the gene. Not perfectly and not completely, but enough to place the animal phylogenetically. Enough to ask which living species produces collagen most similar to that of T-Rex. The answer, published in 2008 by Organ and colleagues in Science, was unambiguous. Among all living animals whose collagen had been sequenced, The closest match to T. rex was not crocodilian. It was not lizard. It was chicken. The molecular signal placed T. rex squarely within the bird lineage, not as a general evolutionary statement about dinosaurs being the ancestors of birds, which had been known for decades from skeletal evidence, but as a specific sequence-level result placing T. rex closer to living birds than to any other group of animals alive today. The second closest match, after chickens, was ostrich. The third was alligator. The crocodilians that had long served as the go-to comparison for theropod biology turned out to be more distantly related, at the molecular level, than the chickens in your grocery store. The bird-dinosaur connection had been building in the fossil record since the 1990s, when feathered theropods from China began arriving in the literature. By 2008, the skeletal evidence was strong enough that the statement, "birds are living dinosaurs," had become standard in paleontology. What the T. rex collagen added was molecular confirmation, measured, sequence-level confirmation, of where T. rex sat within that relationship. What comparative genomics of living birds has since revealed fills in details the fossils cannot provide on their own. The genomes of modern birds, sequenced in bulk over the past decade, with several landmark projects completing whole genome analyses of hundreds of species simultaneously, carry traces of theropod biology that have been modified but not erased. Genes governing feather development, egg physiology, bone structure, and limb formation all show the deep signature of a lineage running continuously from the common ancestor of theropods to the bird outside your window. Researchers have identified the specific gene switches that regulate growth plate activity, the mechanisms behind rapid bone deposition during the adolescent growth sprint that T. rex underwent, and the regulatory networks that govern egg-laying and nesting behavior. None of these are visible in the fossil record. All of them are readable in modern bird genomes that descend without break from the same lineage that produced T. rex. One research program takes this further. Beginning in the early 2010s, developmental biologist Hans Larsson at McGill University and his collaborators began exploring whether the genetic instructions that built theropod anatomy are still present in bird embryos, switched off rather than deleted. The logic is straightforward. Evolution does not typically discard working genetic machinery. It repurposes it, suppresses it, redirects it. A chicken embryo in early development briefly grows a dinosaur-like snout before the genes that produce a beak take over. It briefly forms a more digit-like limb structure before growth reshapes the wing. These are not ancestral forms reappearing by accident. They are evidence that the developmental switches that built theropod anatomy are still present in the bird genome, still legible, still capable of being activated. Larsson's team successfully induced embryos to grow snout-like structures rather than beaks by suppressing the gene activity that drives beak formation. The resulting embryo was not a dinosaur, but it carried, briefly, an arrangement that its genome has not forgotten how to build. Jack Horner, the paleontologist who served as the scientific advisor for the Jurassic Park films, has been among the most public advocates for what has come to be called the chickenosaurus approach, using gene editing tools to reactivate suppressed developmental pathways in chicken embryos and produce an animal with features closer to its theropod ancestors. A longer tail, functional forelimbs rather than wings, a toothed snout. Horner has been explicit that the goal is not a living T. rex, that is biologically impossible without the genome, which no longer exists, but a living demonstration that the genetic distance between a chicken and a Cretaceous theropod is far smaller than the physical distance between them suggests. The project has attracted serious researchers and serious funding. No chickenosaurus has been produced yet, but the preliminary work on reactivating individual ancestral traits has produced results, and each result has confirmed the same underlying point. The genome of a chicken still contains, in dormant form, instructions it has been carrying since before the extinction of non-avian dinosaurs. What T-Rex DNA would have looked like had it survived can be partially inferred from the molecular clock. By comparing the genomes of living birds and estimating the rate at which mutations accumulate over time, researchers can project backward and sketch how many genetic differences separated T-Rex from the lineage that led to modern birds. The answer is a genome broadly similar to that of a large modern bird, but with a set of differences that produced an animal growing to 14 tons, filling an ecological role that no living bird comes close to occupying, and living in a landscape that has since been covered by the Great Plains of North America. The broad strokes of that genome are reconstructible from its living descendants. The specifics are gone. What the 2005 Schweitzer discovery ultimately confirmed is something the skeletal record had been suggesting for 30 years. T-Rex was a bird. A very large, flightless, feathered bird, with lips rather than exposed teeth, a brain tuned for smell and depth perception, and a life history that put it through two completely different body plans before adulthood.